I'm Art Motozenik, and I'm with the American Association of Pharmaceutical Scientists. It's my pleasure to serve as a host for a series of interviews of distinguished pharmaceutical scientists who have made significant contributions to our profession. Speaking on behalf of the Distance Learning Committee of the AAPS, it is our intent to try to reflect on the motivation, inspiration, and some of the actual experiences that develop the early, mid, and later careers of our distinguished scientists. We'd like to explore the perspectives that they have on pharmaceutical scientists and actually look forward to see where they predict the future of pharmaceutical sciences may be going. Today is our pleasure to visit with Malcolm Rowland. Malcolm Rowland is a professor emeritus and a former dean at the University of Manchester School of Pharmacy in the United Kingdom. It's my personal pleasure to interview him because he's a close friend and colleague of many years. So Malcolm, we're looking forward to an opportunity to get inside your analysis of your career. I'd like to begin by asking you about your professional training and background and maybe how you were motivated to go into the academic programs you were at the university level.
Yes, I, as you know, did a degree in pharmacy, and I decided in those days you did an apprentice system. And then you went on to university afterwards, which now doesn't exist. And so I did that apprenticeship and I enjoyed it and decided then to go on to get the degree into pharmacy. And I did very well in academically, and I was already therefore motivated to learn more, just inquisitive. And then I went and did work for 1 year with Pfizer in formulation, actually, which is what I specialized in my final year at university. And it's then that I realized that if you were going to go anywhere in life, it was important that you did actually get a higher qualification, that the professional degree was useful for pharmacy, but if you really wanted to progress. And it's when I went back into the university system and really did my own research, started research myself on things which hadn't been essentially discovered before, that you really began to get that passion they could do. And it was during that period of time that I think I really made a very clear decision to go into academia. And I've never thought about changing that at any time since that time. So I think that's really how it all started.
Well, you've had a remarkable career in not only being successful in producing significant research and publications in the pharmaceutical research area, but also in paving and forging new areas of research. I was curious about early on, were there any unusual inspirations you might have had from teachers or mentors or role models, even as a student, that kind of moved you toward the research areas?
That's very interesting. My professor was a person by the name of Arnold Beckett. He's essentially a medicinal chemist, but a very live wire. And Arnold was one of those people that used to come to you, and if he had something you'd like to do, he'd ask you, he'd really say, would you like, this is something I think is very important, he'd get very excited. And I learned very quickly you didn't take too much notice of that. But what it does, it made you think about what it is you wanted to do. And over that period of about 3 years in which I did my research, I really began to realize that it's you that really decides what needs to be done. You read the literature, you interpret the literature, and you are the one who is going to make the difference. So in that sense, there was at that early stage someone who made me realize that I'm not just going to be told what to do, but actually create the ideas for myself. I think those were the beginnings of that kind of degree of independence, I would say, that one learned. I think prior to that, my background was living with a school teacher. Sounds a bit bad, but I went into a foster home, and the foster parents I had happened to be a school teacher. Mm-hmm. And he was very passionate about learning. And I think that also made a lot of difference very early on in my life, of realizing that.
And who was that person?
The person was, I called him Mr. Brody, was
yes.
the person I
So
went
Mr.
with.
Brody and Professor Beckett were key inspirations for you. Of course, I know Arnold Beckett very well personally as well, and I share your attitude about his independent personality, and I suspect that part of those qualities were transferred into your own outset, because you certainly have that independent look. Did you feel that there were other folks that were influential, maybe not personal context, but people who inspired you from their work or from their literature as the references?
Yes, I think one of them, I did my PhD looking at the pharmacokinetics of amphetamines. I was always interested in quantitative work. I was always interested in mathematics. And there was work being done by Einol Nelson at the time. I don't know if you remember Einol Nelson.
Yes, I know.
And he was working at the time in the area of kinetics, and was very influential in the fact that here was someone who was really in my field, but he looked such a giant at the time, and all I could do was read his publications. The other one, which I forgot about, was that when I was doing my PhD, I met a person by the name of Eckhard Kruger-Thieme. And Eckhard Kruger-Thieme was a German who during the war was imprisoned by the Russians, learnt mathematics. He was medically qualified. But he invited me to go to Borstel in Germany and spend some time with him doing modeling of data. And you can imagine in those days the computers were not very fast, and we would go into these huge big machines and do our work. And that was really also influenced me in terms of the movement towards kinetics and realizing that here was a subject matter that was really close to my heart. That was a combination of chemistry, it was a combination of mathematics, and it was quantitative, and it had a potential for discipline within that. And I think those were the days. And then, of course, there was the publications of John Wagner and Ed Garrett. These were people. So these were people external. I've never met them at that time, but these were people that I really felt were giants in their area at the time through their literature, and that made a strong impact.
Well, that's very good, because I do recognize that your core competence is in pharmacokinetics. And you just shared with us the overview of the dawning, really, of the pharmacokinetic era.
Yes.
Because you just mentioned giants in the field, and these were professional colleagues who eventually you got to meet some of them. And I know Nelson, I met him only once, but he died rather young in his career.
Yes.
But his influence was very strongly felt.
Yes.
Now, you did education in the UK for a long time, and then you did some postdoctoral work, and you had some sabbaticals and appointments in the United States as well. Could you share some of those experiences with us?
Yes, that was interesting. Troy Daniels, who was Dean of the School of Pharmacy, University of California, San Francisco, was visiting the School of Pharmacy in London, where I was doing my PhD, and was going around the labs. And I was introduced to him by Arnold Beckett. And Troy said, "What are you going to do?" So I said, "Well, I'd really like to do a postdoc." And he said, "I've got just the man for you." He said, "Sid Riggleman is in my school. He is in kinetics, and I'll have a word and do that." And that really changed my life. I think there's no question about it. That I met Sid then in a meeting in Edinburgh. It was a British Pharmaceutical conference, and I had never met him before, but there was some question time going on in one of the papers, and a gentleman got up and said, "My name's Sid Riggleman," and that was the first time that I met this enormous person. We both know him, Art.
Very well.
Enormous, wonderful guy, who also had a passion, love for the subject matter, and for life. I mean, he really was a remarkable individual. And so I went to San Francisco initially for 1 year. I just got married. I got married 3 days before that we were due to take up that appointment. And
where did you meet your wife, incidentally?
I met her at a charity dance.
I see.
Those were very common in those days. I don't know if they are now, but we met at a charity dance.
So she's been one of your professional partners all your life.
Absolutely. And in fact, she was the one who typed up my thesis. And
there you go.
And the old stenograph that you just turned over, and Romeo that you did, made your thesis. I remember doing that. So we went over there in 1965, January, and we intended to go for 1 year. But the environment was so lovely. And there's wonderful scientists and wonderful people in that environment, very competitive. You couldn't live in that environment unless you were of a competitive nature. Just it's not possible to succeed. But I really enjoyed it, and I think that was really the greatest changing point in my life, was to be in that environment and to realize that there was a depth and a quality of science, not just in pharmacy, but in the institution itself, that was available to you, and that people were so open and so willing to discuss things with you. And when you're a postdoc, one of the wonderful things is you've got the freedom. You have no responsibilities to anybody. Somebody else is providing you with money, and then you're given the chance to do with guidance, but chance to do what you want. And I think those were really the change. I think Sid was definitely pivotal in that, and the environment of the school. And as you know, Jerry Goyen took over from Troy Daniels to that appointment, and Jerry had built up a very fine school of pharmacy there. So that was a joy and a pleasure.
Well, you've referenced several clusters of where pharmacokinetics was emerging in a pivotal way. Your own work, I might say, took kinetics more into from the exploratory areas of where we're defining maybe even the original models and setting the original equations, to examining elimination and pharmacodynamic correlations and so forth. How did you decide, if you did, to specialize in this area, because it was emerging as very significant? I might parenthetically say, probably when you and I were undergraduates in pharmacy, there was no course in pharmacokinetics. It came about during our time.
Correct. I think it was basically a mathematical subject at the time. I think there's a number of things that worked together to change it, and one was analytical methodologies were becoming available, allowed you to quantify compounds, drugs, following their administration, and that was not available really before. So we suddenly were faced with data. And the second thing was the realization that this was not a mathematical subject, but was really a quantitative subject based on physiology, based on chemistry. And that if we were going to able to move this forward, then we'd have to be far more physiologically thinking in the way we interpreted data. And I think that was, to me, one of the real big changes. And I think if I was to say anything about the subject matter, I think we became much more physiological, much more mechanistic, rather than descriptive. And I think that has really been one of the great changes. And I think suddenly people realized that this was not just mathematics, but this had something to say about the way in which we design, develop, and use drugs. I think that's really what it was.
Mm-hmm. I'd like to switch a little bit to your mid-career, where you began to be significantly a contributor to the pharmaceutical literature. And then later I'd like to explore your teaching aspects. But I knew you were teaching even at that time and began a series of contributions through your students. But how did you transition into intensive research at that time, and where were your major opportunities presented to you?
Yes, that's very interesting. Parallel to the time that I arrived in San Francisco, the Department of Clinical Pharmacology within the University of California, San Francisco, was really becoming well established under Ken Melman. And Ken was one of these real guys. He came across almost like a bear. You know, he could hug you, but he was a squash player, and he was really muscular, but piercing eyes and a lovely broad grin. And he, I think, linked with, I don't know the details, but linked with Sid Riggleman, and decided to set up a program under the IGMS scheme on clinical pharmacology. And I was a young guy, and Sid says to me, here's an opportunity, you know, to get involved. And I then started interacting with medical people and with clinical pharmacology, and a very fine caliber of people. And the fellows that came in were first class in there that they recruited. And I think that made enormous difference to the potential that one had about the subject matter, because here was a group of people who understood what you were saying, but also had something else to offer from the clinical side, from the medical side, so you could get a much broader perspective about the application of the subject matter. And I was very fortunate in that being there at the right time as a young investigator and being associated with that. I think those were very formative, and Sid was extremely supportive of that, that development, and that really grew into a major center of pharmacokinetics and pharmacodynamic research, as you know.
Well, one of the things you're very well recognized for, Malcolm, is the ability to help prepare scientists for careers in pharmacokinetics. So commenting as you did about recognizing yourself the importance of physiology and clinical science, and as we were not trained in those areas, but it evolved, you were very important in contributing not only in your teaching, but in your books and publications and some pivotal texts a classical formula for preparing yourself in pharmacokinetics and examinations of drug metabolism and investigations. How did that evolve? Did you see that role as a rewarding and satisfying part of your career?
That's interesting. My primary interest, I think, and motivation of going into academia was actually the research, and not really the teaching, I think, initially. It's only when you have students who are particularly motivated students that you really recognize the real importance of actually education and training there. I think that the honing down of the methodologies of communication and teaching actually were based primarily at the undergraduate level. And I was very fortunate, and there was several things happening at the time. One was Tom Tozer, who, as you know, is the co-author of the textbook that we wrote in clinical pharmacokinetics, the concepts and applications, that book, was a graduate from the University of California, from the school, but had gone to NIH, and he'd just come back. He'd done his Ph.D. in electron spin resonance. He'd gone to NIH and worked in B.B. Brody's lab, and who was very eminent at the time, in neuropharmacology. And he had come back also as a young faculty member, and we were assigned the responsibility to teach pharmacokinetics of there. And that's when I think that I really, I think about 3 or 4 years, really began to realize how to teach. That didn't come, I think when you're very young, you want to impress, and you start using languages and forms of communication with the student really many times doesn't relate to. Mm-hmm. And I realized that there was a way of communicating to students, both in terms of the motivation that you have, but also the subject matter. And I think over that period of time, there was no textbook that one could really refer to. And so one was really formulating all the time, how were you going to teach this course? And I think that's what made a difference. And then I was very fortunate to have some excellent first graduate students, and I said clinical pharmacology fellows. And they were really a joy. I mean, absolute joy to teach. They were just, you just had to say a few things and they were off. And that's really fantastic. And they were pushing you all the time. So I was very fortunate.
Would you be able to segment some of the key contributions in your research program, either from a chronological pattern or from an application area, that number one, were probably the most satisfying to you, and then secondly, maybe the most challenging to you?
I suppose anyone who goes into academia and moving through and forward in the subject matter tries to develop a sound understanding of what they're interested in. And I would say of all the areas that I really got involved with and sustained in was the area of clearance, the concept of clearance, and what does it really mean, what influences it, how do we use that information in drug design, how do we use that information in drug administration. And I think if you look through all the publications I've done, then I think that clearance idea I think was the one that really was a sustaining thing. And I think in many ways, I suspect, as a concept, was the most impactful of all the things that I've done.
And how about maybe an area where you still have great aspirations and a challenge that you'd see opportunities in the areas that you've worked in, or maybe a sense that other workers might be in?
Yeah, as I mentioned, the impact, I think, and the clearance, that concept, I think that really was I wouldn't call it seminal almost in the way, would be that it was a realization that this was things that could make a difference in the way we understood drugs. But then what I got very interested in is in the broader picture of, could we explain the way drugs were handled by the body rather than describe it? I think that was the change. So I think much of the second part of my research career was what I would call whole body physiologically based prediction. Could we predict how a human being would handle the drug prior to ever giving the drug to the individual? Could we do that? Could we become much more mechanistic predictive from in vitro, from in silico, from preclinical data? And I think that much of my research and my passion, I think over the last 10, 15 years, has really been directed to that subject matter. so that we don't have to use all these animals, which really for drugs that are, for compounds that are never going to come to human use. Why are we doing that? Why are we giving to humans in a phase 1 study drugs which are really going to, you can predict a priori, are going to be not very desirable pharmacokinetic properties? Didn't seem to me that, this day and age we should be doing that. And I think that's the one that I have been very interested in doing. And I think it's the one that I think that there's still a lot of work to be done, but I'm quite convinced in my own way that that's the way it's going to go. We'll predict.
Well, indeed, we do recognize the seminal influence of the work that you speak of, the concepts of clearance. They were very pivotal, for example, in providing a stronger platform of intelligent design for drug delivery systems.
Yes.
And the concept of dose sparing and giving opportunities for patients to be able to seek remedies with minimal drug dosing. So this is a very important area. If we were to reflect on, in your opinion, where the challenges are today as opposed to the ones that of the career path you had, where would you see the emerging areas of today? And then I'm going to ask you also to look to the future. So if you could answer that in 2 parts, what the challenges are today and what you would see in the near future.
Well, within the context of my own subject area?
Of your own work area.
Of my own area, as I said, I think where we're at the moment is we are at the edge of becoming predictive, mechanistically predictive of the way drugs are going to be handled, based upon our understanding of their chemistry and their structure and their physical chemical properties, and linking the in vitro methodologies as well as the in silico methodologies to integrate all of that into one broad picture, essentially, an integrated picture of the way drug is going to handle. So pivotal, the 2 aspects I see, one is a better understanding of physiology and pathophysiology and the disease process that we can describe in a mechanistic way, quantifiable mechanistic way. And the other one is a better understanding of how drugs interact with the biological system to manifest the way that they behave. And I think those 2 things are coming together. And when they come together, you're going to see a quantum change in the way in which we'll think about drugs. Because we'll be able to be closer to what you see in the aerospace industry, where we'll be able to say, this is the way we expect this drug to be actually handled by the body, even before we've given it. And therefore help the medicinal chemist to actually design the right sort of molecules that are going to have those properties. And I don't think we're there yet. It's a lot of empiricism still going on. But I do believe that's the direction we're going to go. And that's what I think a lot of effort is going in at the moment. If you ask me, where the future is, then I would say that this subject matter would nestle within the broader range of what people would commonly call now systems biology. That is, that it's just one component, right, of a much broader picture, and that is how do we operate as a biological system? What is the way in which the genome and the interaction with the environment and with aging, how does that actually manifest in the behavior, the characteristics, or what we would call the phenotypic characteristics of individuals? And how does that understanding allow us to design better molecules? And also, how do we then know how to administer them in a much more optimal manner than what we're doing? And I think that's going to be the direction that I see it going.
Well, that's most interesting. You remarked in several of your comments about the extension to new disciplines, multidiscipline, and even in your career you've certainly demonstrated that. Where would you see some of the companion disciplines that we're reaching out for? You talked about, for example, adding in silico design to the traditional in vitro, in vivo correlations and animal and human correlations that we've done. Would you see any new opportunities for the pharmaceutical sciences to stretch and grow?
Yeah. There must be many areas. But if I look at the areas which are going on, for example, in drug metabolism, I think where we are at the moment is that we can say, with looking at a structure of a molecule and its properties, we can say that it's likely to be metabolized extensively or not. We can say even what the enzyme that's going to be probably predominant, and even some of the pathways. But what we're not quite there yet is the quantitative side. What's going to be the rate of that reaction? And instead of just experimentally doing it, can we actually predict it ab initio? And I think there's a whole lot of understanding of the nature of the interaction of drug molecules with biological systems to predict their functionality in a quantitative way. And I think that's going to happen. in the future too. And I think there's enormous potential within those areas to really become much more mechanistic, much more quantitative in that subject area, so that they'll be the components of the larger system model.
Right. You have made significant contributions not only in the UK and the United States, but you certainly are well recognized globally as a major contributor in pharmaceutical sciences, Malcolm. How would you have reflected on your interest and zeal, because you've been most generous in sharing your appetite for pharmacokinetic research and for teaching and contributing at conferences in international meetings? What kind of challenge do you see in shrinking the global research community, and what have been your reflections on your past experiences?
Yes, as you know, I mean, my view of that we're all human beings within the globe, and that really the differences between one individual and another is really more of a quantitative rather than qualitative nature, and therefore the rules that we're talking about should apply to everybody. But I've been very interested, from the point of view of a scientist, to put back into the community those things that I am passionate about. And when I returned to the UK, I tried to get involved in national and also try to bring the European scene into one sort of common umbrella. And I spent a large part of the last 10 years of my life really engaging the pharmaceutical sciences community, particularly in Europe, and trying to bring it together under one umbrella, and then linking in with both the North American continent and particularly the Japanese community to try to develop global type of structures to communicate ideas and bring things forward on that. And that's been a great pleasure. It's a lot of work, very interesting to get involved with that dimension. So I was heavily involved in the establishment of the European Federation of Pharmaceutical Sciences in Europe, which is now, I think, well on its way and having influence, and also linking in with the AAPS, for example. I come as a regular member to the AAPS, and I've always done that, and I will continue to do that. And also Japan and other things. And I do see that what's now going to be critical is to engage in with the emerging countries, the China, India, and these other countries, to not only in terms of outreach programs, I think very important, but also to realize that they are going to be your fellow players in the future in the pharmaceutical sciences, and we should really try to nurture that at an early stage so they will see themselves as a global player in that community too. I think that's going to be very important to us.
That's very good. In reflecting on the significance of clinical results and clinical data, quantitative as you described it, and just looking at the attempts at governments to regulate and overview the safety and efficacy of drugs, how have you seen the role of regulatory science using the kind of scientific research and disciplines that you introduced with your colleagues?
Yes, that's a very important part. I think one of the most important, because all the science, all the things you can discuss, all the concepts, which can, I think there's no doubt, had an influence in the actual development of new pharmaceuticals, but also in the use of the compounds. That's all come through. But if you don't engage the regulatory people and understand the changing science and work with them to change it, then what happens is you get a kind of a stagnancy developing at that level, which hinders the bringing to the society of drugs for unmet medical needs. And so I think that's been a very, very important part of the process. And I think that's been, very successful in the United States. I think more successful than anywhere else I know of. But there is moves also within Europe, for example, to engage the regulatory authorities in discussions about the impact of science and how we can move together to make better guidances about drug use, better appreciation of the issues involved, and how can we work with both the industrial, academic and regulatory communities to know what are the stumbling blocks in that system and how can we actually creatively and innovatively change those so that we can bring forward the medicines to the patients. I think that's very important.
Well, thank you. You are a much respected voice in the regulatory community, and I think probably a good content expert. Where would you see some of the challenges to use more clinically based pharmacokinetic science in the regulatory area that you would hope to see achieved in the next few years or in the decade ahead?
Yes, I see that primarily in the way we'll be thinking about how we'll integrate information in the phase 1, phase 2, phase 3 area. Instead of seeing them as boxes, we see them increasingly as continuums. And I think these labels are really kind of a bit misleading. I think the better way, and I think a very dear colleague of mine who who also had a strong influence that I should not forget, is Louis Scheiner. Louis was a wonderful person. He unfortunately died last year. A very sad loss to the community. A giant in his own field and he was a person that I taught with for many years. And he's always brought up this idea that really when we're going forward in drug development, there are two basic components. One is the learning part, and the other one is the confirming part. And that much of what we're doing in drug development is actually the learning part. And we should be open, flexible in all of those parts of integrating. And only at the end, when we think we've got sufficient understanding of a compound, should we go on to the confirmatory phase 3 studies. And I think that's absolutely true. And I think that understanding that these things are continuums and actually continue from the preclinical to the clinical, not just that one, as you know, Art, is a very important part.
Yes.
If we don't get it right at the very early stage, then all the beautiful development in the world doesn't help you in bringing the right sort of molecules to it. So Louis, I think, was the one that really, and he and I used to spend a lot of time together during the courses that we taught together for 20 years, really philosophizing about a number of these, what I call basic but impactful issues that will affect the way in which we think about drugs.
Could you reflect on what probably were some of your most satisfying achievements, maybe a research area or a cluster of students that made a significant impact as well in your career?
That's a difficult question. Think of that. As I told you, I think in terms of a cluster of people, I think there was a cluster of fellows and Ph.D. students in my very early days that really make things. Lewis was one that I think, I'll probably forget people, but there was Terry Blaschke that was there. Roger Williams was a fellow, as you know now, who leads the USP Pharmacopoeia. There was René Levy, who was a graduate student of mine, who is now at Washington, has done wonderful things. There was a whole group of people in that area that were just a joy to interact with as well. And as you know, and the other one, there's two other players in addition to Sid, that I mentioned before, who was very thing, and that was Les Bennett came on the scene at this thing, and he and I did a lot of interaction together, and the great interaction that's lasted the years. And the other one was Tom Tozer, who was there. So I think that was, I say, the greatest thing, and the pleasure of seeing that idea spinning off. And I think that happens in life. You see that whether it's in, suddenly the Beatles get together, for example, as a group of people together, or somebody gets together in science, or it could be in any sphere, or art, where there's a group of people suddenly get together and things go. And I think those were our thing. And anybody that spun out of there, I think, felt that. And I think got that. That was very, very important.
Well, you certainly remarked about some excellent synergies you've had in your career path. Those are good examples. Well, indeed, Malcolm, I think I'd be remiss if I didn't ask you to comment in talking about the seminal influence of your work, the significant impact that the book that you and Tom Tozer have put together is really a classical text today. How do you feel about the evolution of that book and its revisions and the impact it has in education today?
Yes, it was very interesting how that came. I had talked previously about the idea that when we were starting to teach the area of pharmacokinetics, Tom and I were made responsible for teaching that course within the University of California School of Pharmacy. There was no textbook. And I remembered quite vividly, we used to kind of do the exams the night before. We used to write them, type them, and one of us used to spin essentially a coin of who's going to wait up doing the Roneo and have it ready for the class the next day. So that was there. And I said to Tom one night, it was very early hours of the morning, I said, this is crazy. We are actually working our butt off. We've got no textbook that we've got the student to go to, and we are trying to always decide where we're going to get our questions from and so forth. So really we should write a textbook. And that was the idea that came about there. And subsequently, when I was on sabbatical in London, we decided to do it with those period, and we started there in about 1973 to actually put our ideas down on paper, because we had formulated a course which was physiologically oriented, which kept the maths in perspective. And then what we did was we started writing this book. And over the next about 5-7 years, that book evolved to the first edition, which was of the book Clinical Pharmacokinetics, was in 1980. It was a fairly slim book, and we had intended to actually write a companion book, and we made reference to that in the preface of the first book. But then what happened was, when we decided, it became quite successful very early on, and we decided then to write the second edition of it. We decided that really what we do is incorporate the ideas for the second book into one larger book. And I think that was a very wise decision. And by that time, this book was now becoming adopted not only in schools of pharmacy within the United States, but we understood, but in Australia and in parts of Europe, it was beginning to be adopted. And the second edition, I think, was a major jump in terms of its content. And from then on, and the third edition, I think, was a further improvement. And so now, if I understand it from talking to the publishers, it's the textbook of the subject throughout the world that students go to for learning about pharmacokinetics. So I think that one, and it was one of those times, I think you know yourself, was where you come across an individual where the chemistry works. There are no barriers. There's nothing that stops you. You can say what you like. You can shout at them. You can really argue with them. But that's at a professional level. But at a human level, you are just solid friends. There is nothing going to disturb that relationship. And that was Tom and I. Tom and I just, hand and glove, just worked together beautifully. And that's continued even today. As you know, we're now writing our fourth edition. We've now decided to write another introductory textbook on kinetics and dynamics, pharmacokinetics and pharmacodynamics, because we feel passionately that this is a way to communicate to a much, much wider audience than you could ever achieve by teaching a particular class.
Right.
And that certainly, I think, proved to be the case.
Well, that's a wonderful insight, Malcolm, because certainly your text with Tom Tozer is a legacy. But the insight you just shared with us about producing such a text, there are so many great colleagues of ours who owed us a text and never really got to it.
Correct.
So for actual implementation and the satisfaction of the execution of that, it's a wonderful achievement. We congratulate you on that.
Thank you.
There's one other area that you've been very, very acknowledged for, and that is the introduction of the workshop concept for a lot of interdisciplinary scientific meetings. Maybe you might share some of the efforts that went into the development of that and how that evolved.
Yes. As you know, Art, after 10 years at San Francisco, I decided to go back to the UK, where I was born, for a number of different reasons. But one of them was that I felt that there was a golden opportunity and a need within Europe for the subject of pharmacokinetics developed. Despite the fact that the origin of the word pharmacokinetics was coined by Dost, a German physician, in Die Blutspiegel was the book that he put out. And despite the fact that Eco Kubo-Thieme was seminal in propagating those ideas, which came from Germany, the fact remains is that it essentially was not taken up in any serious way in Europe, in contrast to the North American continent. And I felt that that was something that needed to be done. And the other thing to understand the contrast between the European, at least the UK model, and the US model is they don't have formal postgraduate education. So if a person, as you mentioned before, didn't have undergraduate pharmacokinetics, then they would never have been exposed to it and understood it. Mm-hmm. So what we had was a situation where people, particularly in industry, who were realizing that they needed to know pharmacokinetics because that was an integral part of the drug selection, drug development process, had nowhere to go to find out where to learn the subject, particularly in Europe. And so I decided when I got back that one of the things I would do was disseminate our ideas. And I invited Tom, particularly at the early stages, to develop workshops. And they just took off like a flash. Mm-hmm. I mean, they really did. And before we knew what we were doing ones in UK, in France, and in Germany, and so forth. And that started off in about the late 70s. I returned in 75, and in 77 we started the first one. And as I said, we didn't stop from then on. And they are still going every year, right, full, since at that time. We thought that maybe 3 years we would teach everybody need to be taught. the subject, how naive we were.
And you've also set a pattern of precedent for other people to do workshops in a successful manner that you have done
Correct.
as well.
The second one, if I can just think, was Louis Shiner. Because Louis and I was on faculty at the School of Pharmacy and he was a fellow. But that really wasn't a true representation of our relationships. We were just two young people together. He comes from medicine, very interested in trying to understand data and how to interpret and analyze data. And I was very interested in why are drugs handled the way they are. So we came from different backgrounds. And Louis came to me one day and said, you know, why don't we teach a course together? I, with my knowledge and subject matter of data analysis and statistics and of the pharmacodynamic side, you from the kinetic side. And we formulated this workshop on advanced methods in pharmacokinetics and pharmacodynamics. And that, I think, was a unique course in its own right. And while I've decided to step down from teaching it last year when Louis died, I'm delighted that it's being continued by the tutors of those courses. And I'm sure it'll go from strength to strength.
Well, in terms of continuity, I'd like to at least solicit from you an observation about what kind of advice you might offer to young women and men entering the pharmaceutical scientific professions today.
Well, I come from an academic career, so I see it in that perspective. I think that one of the things that I felt always very important is that you have to be passionate about the subject matter. It's no good going into a position because you believe that you're going to have a solid career and that you're really going to, it's going to be a stable thing, an income base. I think that's important. But when you're young, you've got to have that passion and you've got to believe that you have something to offer in it. I think that's critical about it. And if you don't have that, I just don't think you're going to get very far. And if you want to go into academia, which is what I've done for most of my life, then what you then have to do is you have to say, what is the mark that I wish to make? What is the area that I wish to pursue? And you've got to have, at a minimum, a rolling 5-year program in your mind. It's no good kind of running around and doing a lot of things. If you really want to make a mark, you've really got to have a commitment, and you've got to see that commitment through. And there may be times when things are hard and grant money is not coming in so easily. But if you really think it's important, then think. And the other thing is you've got to be in the right environment. The chemistry, the right environment, is critical to that young person developing. And that's what I would say from my academic perspective that I think are important ingredients.
Well, that's well said. I'm sure it'll be well received. So indeed, with all the many influences and the time and energy requirements you had, were there any other pivotal people in your life who helped motivate or inspire you, maybe even during times when things were challenging and difficult?
Yeah, I mean, I should not forget, my wife. As I mentioned very early on, we got married just a few days before we departed, actually on the New York, the Queen Mary. And we went across the Atlantic, 5 days honeymoon on that period. My wife, Dawn, because she's not in science. She's an artist, sculptor. But, you know yourself, Art, that in this type of career, you get a passion for something and you dedicate your life. You come home late at night after night because of your research. You're out on the circuit in the sense of going around talking to different communities. It's a global arena. So that could never have been possible. And I could never be the way I am and really feel I'm very fulfilled, but simply very happy about life. If it wasn't for that, I had a great wife, and she's been supportive throughout that time. She listens to all my problems, etc., even though she can't help me. She's always there. Yeah. And I think without her, I don't think I would have really done what I've done. I'm quite sure about that.
Well, I'm sure that's very important to you. Comment on your dedicated partner in life and your career path. It's very well said.
Thank you.
Well, Malcolm Rowland, distinguished professor, it's been a pleasure interviewing you today, and I congratulate you.
Thank you very, very much. Enjoyed it very much.
Thank you.
Thank you.